Welcome back, deep divers. Have you ever found yourself wondering about the truly complex neurological conditions like um, Parkinson's disease, or even just how we push the very boundaries of understanding the intricate workings of the brain itself? Well, today we are taking a truly deep dive into a groundbreaking piece of technology that could really revolutionize how scientists study the brain. We've got a fascinating set of sources today, mostly centered around this academic paper called A Novel Charge-Neutral Programmable Wireless Brain Stimulation System for Rat Experiments. It's quite a mouthful, but it's impressive work. Comes from a team led by Srinivas Bhaskara and his colleagues. They're at the Indian Institute of Science and Ramaya University of Applied Sciences. So our mission today really is to unpack this innovative research. We'll explore the critical need for, better tools in neuroscience, delve into how this team engineered such a sophisticated solution, and then crucially what its implications are for both understanding and maybe potentially treating brain disorders down the line.
We're talking tiny, wireless devices that can literally spark new discoveries, opening up whole new avenues of research. It's pretty exciting stuff.
Indeed. And this deep dive, it'll highlight not just what they built, which is interesting in itself, but more importantly, why. Why such precise, adaptable, and robust tools are absolutely essential for advancing our understanding of brain function and dysfunction, too. This is particularly relevant, in the context of conditions where current clinical treatments, well, they still have significant limitations. There's a real demand for continuous innovation and exploration.
Okay, let's unpack this then. Many of us are, I think, at least somewhat familiar with deep brain stimulation, DBS, right, as a therapy for Parkinson's disease. It's been around for decades, clinically accepted, especially for folks experiencing those severe motor symptoms that maybe don't respond so well to medication anymore. It's truly life-changing for many people. It offers a new lease on life for individuals whose tremors or rigidity, severely limit their daily activities. But the question we need to ask is it perfect? Or could it be even better?
That's a really important question, and it gets right to the heart of why this kind of research is so vital. While DBS of regions like the subthalamic nucleus, the STN, or the globus pallidum internum, the GPI, while that is indeed an accepted clinical practice globally, the outcomes are often described, even in the scientific literature, as unsatisfactory in some aspects.
Unsatisfactory? Like how?
Well, for example, we're talking about incredibly debilitating conditions like gait freezing. That's where a patient can suddenly feel like their feet are just glued to the floor. It often leads to falls, severely impacting their mobility and, their independence. Or issues with speech, something called dysarthria, which can make communication incredibly difficult. Or even certain non-motor symptoms like depression or cognitive issues where, frankly, there's still much to improve upon for patients' overall quality of life.
Wow, okay. So it helps, but it doesn't solve everything.
Exactly. This means that research is actively ongoing. People are exploring other potential brain regions beyond those traditional targets, and also refining the existing stimulation protocols to try and achieve better, more comprehensive therapeutic effects. We're looking for ways to fine-tune these therapies, essentially, to make them even more precise and effective for a broader range of those debilitating symptoms.
So if we want to improve these human therapies, we want to maximize the benefit for patients, we fundamentally need to understand the brain better. Makes sense. And for that, researchers often turn to animal models, particularly rats, to conduct this exploratory sort of foundational research, right? They can't just go experimenting directly on human brains, obviously.
Precisely. Rat models are absolutely critical for this type of exploratory research. They allow for controlled experiments, identifying new targets, testing novel stimulation patterns, observing behavioral outcomes. You can do all this in a way that just isn't feasible or ethical in human trials. For instance, when studying Parkinson's, you might induce Parkinsonian symptoms in a rat model, then try different stimulation parameters, see how they impact motor control or gait or even cognitive function. However, and this is a significant but, the tools that have historically been available for these crucial animal experiments, well, they often present significant hurdles. Hurdles that can limit the scope, the accuracy, and ultimately the clinical relevance of the research itself.
Okay, now this is where it gets really fascinating. What were the specific concrete problems with those existing brain stimulators that this particular team was trying to solve? It sounds like a pretty niche problem maybe in the lab, but with potentially huge implications for human health down the road. What was holding researchers back?
It truly is a niche problem with broad impact. The limitations of previous systems were, well, numerous, and they significantly constrained the research people could do. Okay, first, just consider the sheer size and weight of commercially available systems. They are typically far too bulky and, frankly, too costly for use with small animal models like rats.
Right.
Imagine trying to study a rat's natural gait or explore complex behaviors, like how it navigates a maze or interacts socially, if it's burdened by a large, heavy device strapped to it.
Yeah, you wouldn't get natural behavior at all.
Exactly. It's simply impossible to obtain valid data on natural movement if the animal's behavior is overtly affected by the equipment itself, rather than by the brain stimulation you're actually trying to study.
Okay, so size was one big issue. What else?
Building on that, many older systems were tethered or wired.
Ah, wires. Always the problem.
Right. This meant the animal's movement was severely restricted by a physical cable connecting it to external equipment. So, if you're trying to understand how brain stimulation impacts free-moving behavior, for instance, how it improves a rat's ability to initiate movement or maintain balance, which are direct parallels to human Parkinson's symptoms, well, a wire physically connecting the animal completely compromises the experiment. The entire premise of studying natural behavior, which is essential for translating findings to humans, is just undercut. It's like trying to study a bird's flight by keeping it on a leash.
Okay, got it. No wires. What else?
Then there was the critical issue of lack of programmability. Early customized stimulators, like some developed by Ewing et al. Or Kuzani et al., often used what's called passive charge balancing. Now, while these were custom-built, their critical drawback was that the stimulation parameters, things like current amplitude, frequency, or pulse duration, they could not be changed after the device was fabricated.
Really? So you were stuck with whatever settings you built it with.
Exactly. For exploratory research where the exact optimal parameters for a particular brain region or a specific therapeutic effect aren't known beforehand and often need to be discovered through trial and error, this is a massive disadvantage. It forces researchers to build multiple distinct devices for different settings, which is incredibly time-consuming, expensive, and just slows down the whole pace of discovery.
Okay, that sounds incredibly frustrating.
And even when some systems offered some programmability, there was still difficulty in real-time adjustment. For instance, a system by Fleury et al. It required physical disconnection from the subject, reprogramming via a PC, and then reconnection.
Oh, wow. So you'd have to stop everything.
Think about that in a live experiment. You observe a subtle change in the animal's behavior, and you want to immediately tweak a parameter like the current strength. With such a system, you'd have to stop the experiment, unhook the animal, wait for the device to be reprogrammed, hook it back up, and hope the animal's state hasn't changed in the meantime. It's incredibly cumbersome and highly impractical for dynamic, responsive research.
Yeah, that sounds like a nightmare workflow.
Then, another significant limitation was single output capability. Acosta et al. Developed a biphasic current stimulator for rodents, but it could only generate one current output at a time.
So only stimulate one spot.
Right, to stimulate multiple brain regions simultaneously, which is often required in complex neurological studies to understand how different areas interact in a network, or maybe to achieve more comprehensive therapeutic effect. This is a significant bottleneck. Achieving multi-region stimulation with such a device would demand high hardware, area, and power consumption for multiple individual units. Just impractical for a small, freely moving animal.
Okay, and you mentioned electrodes too.
Yes. Furthermore, some real-time programmable devices, like one by Ramscher et al., faced limitations in the number of electrodes that could be interfaced due to their specific architectural design. Modern neuroscience often requires the ability to stimulate across several points or regions to fully map or modulate brain activity, needing more than just a couple of electrodes.
Right, the brain's complicated, you need more points of contact.
And finally, a truly critical point, often overlooked by, the layperson, is the issue of charge imbalance and potential tissue damage. Biphasic pulses that go both positive and negative, are universally preferred over monophasic stimulation in deep brain regions.
Why is that so important? Biphasic versus monophasic?
Because monophasic pulses, which send current in only one direction, can lead to unwanted electrochemical reactions and the gradual accumulation of excess electrical charge in the delicate brain tissue. This accumulation, as highlighted by researchers like Pialet et al., can lead to irreversible tissue damage. It can destroy neurons, cause inflammation, or even scarring. So maintaining what's called charge neutrality, making sure no net excess charge is injected into the brain over a full stimulation cycle is absolutely paramount for the safety of the animal model and for obtaining valid, reproducible experimental outcomes without causing, confounding tissue damage.
Wow. So essentially what you're saying is that researchers desperately needed a device that was tiny, completely wire-free to allow for natural movement. Incredibly flexible in its programming so parameters could be adjusted in real time without interrupting an experiment. Capable of stimulating multiple areas simultaneously to study complex brain networks and, perhaps most importantly, absolutely safe for the delicate brain tissue by maintaining perfect charge neutrality. That sounds like a tremendously tall order, a real engineering Everest.
It absolutely was. They were asking for a lot, but all for very good reasons based on the limitations they were facing.
All right, so the challenge is crystal clear. Researchers were hitting significant roadblocks with the existing technology. Now let's get to the good part. How did this brilliant team at the Indian Institute of Science actually tackle these formidable issues? What did they build, and what are its core, groundbreaking features that set it apart?
What's fascinating here is their incredibly integrated and thoughtful approach to solving these multifaceted problems. They developed what they call a wireless biphasic pulse generator that directly and really quite elegantly addresses every single one of the limitations we just discussed. It's a real testament to clever engineering where they meticulously considered not just the electronics, but the practical realities of working with live animal models.
And when we say wireless, we're talking full freedom of movement for the rat, right?
Okay.
No more restrictive tethers getting in the way of natural behavior studies. That sounds like a game changer.
Exactly. That untethered freedom is absolutely key. It's what allows researchers to study naturalistic behaviors like gait, exploration, or social interaction without the artifacts introduced by physical restraints. Without it, you can't truly understand how brain stimulation impacts complex naturalistic motor or cognitive functions, which is exactly what we need for conditions like Parkinson's. You need the animal to be behaving naturally.
This is where it gets truly exciting for our listeners, I think. Let's talk about the specific details of this device. It's quite remarkable when you break it down. First, that miniature design for rodents. This was a huge hurdle for previous systems, you said. How small are we talking?
Well, this device measures a compact 4.3 centimeters by 3.8 centimeters.
Okay.
To put that in perspective for you, that's roughly the size of a standard matchbox, incredibly small.
Wow.
And the PCB, the printed circuit board itself, weighs a mere 9.89 grams without the battery.
Okay, under 10 grams. Is that light enough?
Yes. Critically, this falls well within the accepted range of 10% of an adult rat's body weight.
Why is that 10% rule so important, though?
That 10% rule is critical because any device weighing more could unduly burden the animal. It could alter its natural movement, its posture, or even its stress levels. If a rat feels uncomfortable or its movement is hampered by a heavy pack, your behavioral data will be compromised. It introduces confounding variables.
Right, you want to study the stimulation, not the heavy backpack.
Precisely. So this lightweight design ensures the stimulator itself doesn't introduce those variables, allowing scientists to observe true, unadulterated behavioral responses to the brain stimulation. And they didn't just stop at making it small, they thought about how it would actually be used. The system is designed to be connected as a backpack on an adult rat.
A backpack.
Yeah, the source even shows images of a tailored rat jacket with a custom 3D printed holder for the stimulator.
Ah, that's brilliant. A little rat backpack.
It is. It's such a clever and practical solution for facilitating undisturbed behavioral studies where the rat can move, eat, and interact as it normally would.
Okay, so size and form factor check. What about the programmability? You said that was a huge limitation before.
Yes, that design consideration for practical deployment is crucial. But beyond the physical form, the real-time programmability is truly a game changer. Unlike those previous systems that had fixed parameters after fabrication, this device allows for real-time programming of critical stimulation parameters.
Real time, meaning while it's running.
Exactly. While the system is deployed and actively stimulating, this means researchers can adjust the current amplitude, varying it precisely from 0 up to 2.04 milliamperes, or change the frequency and even select which specific electrode pair is active, all wirelessly.
So you could be watching the rat and adjust things instantly.
Precisely. Imagine, you're observing a rat's gait, you see a specific tremor, and you can immediately, wirelessly, increase the stimulation frequency or current and see the effect instantly. This real-time flexibility is invaluable for exploratory research, where optimal parameters are often not known beforehand and are discovered through iterative testing and observation in a live experiment. It drastically speeds up the process of finding effective stimulation protocols.
Okay, that flexibility is huge.
Right.
And then there's the absolutely vital aspect of guaranteed charge neutrality. You mentioned this earlier as a major safety concern, something easily overlooked. How does this system actively address it?
Absolutely. It's a fundamental safety and ethical design principle. The system is built around the generation of those biphasic pulses. This isn't just a technical detail, it's a crucial safety feature to prevent tissue damage. The system employs a clever switching logic where the same network of components is involved in generating both the cathodic, negative, and anodic, positive phases of the pulse.
So the same pathway makes both positive and negative pulses.
Essentially, yes. This helps ensure an equal and opposite flow of current. And critically, the electrodes are actively pulled to ground during every cycle.
Pulled to ground. What does that do?
It actively discharges any tiny residual charge. This mechanism actively maintains charge neutrality, preventing any accumulation of excess charge in the delicate brain tissue, which, as we discussed, could otherwise lead to irreversible damage or confound experimental results. It's like a built-in safety valve that protects the brain during stimulation every single pulse.
Okay, safety covered. And it solves that limitation of only stimulating one area at a time, right? That sounds like a huge step forward for understanding complex brain networks.
Precisely. This device overcomes the single output limitation of prior systems. It uses a clever technique called current mirroring.
Current mirroring.
Yeah, it essentially allows it to generate multiple biphasic pulses simultaneously. In its current design, it produces two distinct outputs, with the clear potential for more based on the components used. This multi-output capability is vital for stimulating and observing the complex interactions between different brain regions, which is often necessary to understand or modulate complex neural circuits involved in behavior or disease.
So you could stimulate, say, two different parts of the motor circuit at once.
Exactly. For instance, you could stimulate a motor region and, at the same time, another area known to modulate that motor activity, providing a much richer understanding of brain function.
That opens up a lot of possibilities. And finally, a huge practical advantage for any research lab, especially those with limited budgets or time: rapid development and cost-effectiveness. This seems often overlooked when discussing advanced tech, but it's really important, isn't it?
Indeed, very important. Instead of relying on application-specific integrated circuits, or ASICs,
Those are the custom chips, right? Expensive and slow to make.
correct. Custom-designed microchips that can take several months for development, fabrication, and rigorous testing, often costing hundreds of thousands of dollars. Instead of that, this system is ingeniously built using commercially available, off-the-shelf, discrete components.
Stuff you can just buy.
Pretty much. This strategic choice allows for a significantly faster turnaround time. The paper highlights less than 2 weeks for fabrication and testing.
2 weeks compared to months. That's incredible.
It really is. This makes it highly accessible for research labs around the world, reducing development costs, minimizing lead times, and just accelerating the pace of scientific discovery. Scientists can get to experimenting much faster.
That speed to deployment alone is a massive benefit for research, letting scientists iterate and test ideas much faster and get their experiments up and running without those long, expensive waits. This truly frees them up to focus on the science itself.
Absolutely. It lowers the barrier to entry for this kind of advanced research.
Okay, now that we understand what they built and why it's so important, let's peel back the layers and look at the how. How did the researchers integrate all these complex requirements? Miniaturization, real-time programmability, charge neutrality, multi-output into such a compact and incredibly effective system. What are the key technological components that make this deep dive possible for, our listeners?
Yeah, this raises an important question for any engineer or scientist, right? How do you build a robust, programmable, and safe device with readily available off-the-shelf components that also needs to be incredibly small? The answer lies in their thoughtful selection and precise integration of specific electronic building blocks, each serving a critical function within the overall architecture. Think of it like a finely tuned orchestra where each instrument plays a specific role to create a complex symphony.
Fascinating. Let's break down the main components then, piece by piece, like the instruments in your orchestra. At the very heart of the system, acting as its brain, is the microcontroller. They chose the CY8C5668AXI LP034 from Infineon Technologies. What makes this particular chip so suitable? And can you simplify some of the jargon like IDAX for us?
Sure. This microcontroller is a low-power workhorse, which is important for battery life, and a key feature is that it's equipped with 4 configurable current digital to analog, or IDAC, converters.
IDAX, digital to analog current.
Exactly. Think of these IDACs as tiny precision digital to current translators. They take a digital command like send 1 milliampere from the program software and convert it into the exact analog electrical current needed for stimulation. These IDACs are crucial because they can be programmed to generate either source or sink currents within a precise range of 0 to 2.04 milliamperes.
And that range is good for stimulating rat brains.
Yes, that current range is specifically chosen because it's known to be effective for stimulating the cortical surface of the brain in rats. The device is programmed using C language via PSoC Creator software, which is essentially the IDE, the integrated development environment, where the digital commands for the current amplitude and frequency originate. This is essentially where the brain gets its instructions.
Okay, so that's the brain. Next, we need power. What about the battery and charge pump? It sounds like there's a clever way to get the voltage they need in such a small package.
Absolutely. For power, a compact 3.7-volt, 500 milliwatt lithium polymer rechargeable battery weighing only 10 grams powers the entire system. A smart design choice is making it user-changeable, allowing researchers to swap in different capacities based on the specific duration or power needs of their experiment. Maybe they need a longer experiment, they can put in a bigger battery.
Right.
To provide a stable and regulated power supply for the rest of the circuitry, especially for generating those biphasic pulses with a higher compliance range, they use a MAX6 682 ESA plus IC from analog devices. This acts as a charge pump.
Charge pump. What does that do exactly?
It's a clever circuit that uses capacitors to essentially pump and boost the voltage from the battery's 3.7V up to a stable 5V, which is the VDD supply voltage they need. Critically, it does this without needing a bulky magnetic coil called an inductor, which is often required in power circuits but would have significantly increased the PCB's size and weight. So it's a key component for miniaturization. It lets them get the voltage they need without adding bulk.
Ah, okay. Clever trick to save space. Now, to achieve that multiple output capability, they rely on matched transistors for current mirroring. Can you explain what these are doing and why they chose the specific type of transistor, MOSFETs?
Right. To generate multiple simultaneous current pulses, they use a component called ALD1107SBL from Advanced Linear Devices. This is an IC, an integrated circuit containing 4 p-channel MOSFETs.
MOSFETs.
Metal oxide semiconductor field effect transistors. They are preferred over what are called bipolar junction transistors, or BJTs, for current mirroring operations because they have essentially zero gate currents.
Zero gate current.
It means almost no current flows into the control terminal of the transistor itself. This leads to a far more precise and stable replication of the current from the IDE. This basic current mirror configuration allows the system to generate multiple biphasic current pulses simultaneously. It essentially takes the current defined by the microcontroller's IDACs and replicates it almost exactly for different stimulation pathways. Crucial for precise multi-region stimulation. While there is a slight mismatch noted, typically 0.5% at 25 cc, it's a known characteristic of these components, and importantly deemed acceptable for the experimental outcomes they're seeking, given the biological context.
Okay, so mirrors the current accurately enough. And for controlling the actual flow of current and creating those critical biphasic pulses, the ones that ensure charge neutrality, they use analog switches. How do these work? Like little traffic controllers for electricity?
That's a good analogy, yeah. They use the MX394 EUP from Analog Devices, which is a quad single-pole double throw, or SPDT, analog switch IC. Think of these SPDT switches like tiny, very fast railway junctions for electricity. They can instantly reroute the current in one direction or the other. Since the system requires 10 SPDT switches for its various routing and polarity reversing functions, essentially deciding which electrode gets the positive current and which gets the -3 of these ICs are utilized. These switches are absolutely key to reversing the direction of current flow to generate the biphasic pulses. They're low on resistance, less than 35 ohms, ensures minimal voltage drop across them so you don't lose power. And any negligible leakage currents, which are in the nanoampere range, are further managed by actively grounding the electrodes between pulses. Another part of that crucial safety feature for charge neutrality.
Got it. And how does the system communicate wirelessly with the user? That's what makes it so dynamic, right? That real-time adjustment.
Exactly. For wireless communication, they integrate an HM10 Bluetooth Low Energy, or BLE, 4.0 CC2541 wireless module.
BLE, so low power Bluetooth.
Right. Bluetooth Low Energy is specifically chosen because it offers efficient, low-power communication over short distances, perfect for a battery-operated device on a freely moving animal. You don't want the communication draining the battery. This module facilitates the wireless communication between the stimulator device itself and the user interface, enabling that remote control and parameter adjustment without any physical tethers.
And that remote control brings us to the graphical user interface, or GUI. This is where the researcher actually interacts with the device sitting at their computer.
Exactly. The GUI is the user's window into the device's functionality. It was developed using MATLAB 2023A. This software interface allows the user to wirelessly search for and connect to the device, select specific electrode pairs for stimulation, and program parameters like current amplitude and frequency in real time. This is the crucial link that makes the device truly programmable and adaptable throughout an experiment. It puts control directly into the researcher's hands without needing to physically handle the animal or device during an ongoing study.
That's a lot of specialized components working together in a tiny space to achieve such precise control. But how does the actual sequence of events unfold to generate those specific biphasic pulses that keep the brain safe while stimulating it? It sounds like a perfectly choreographed electrical dance.
It truly is a meticulously choreographed sequence, yeah. And the precise timing and control of these pulses are absolutely critical, not just for efficacy, but, as we discussed, for safety. Let's walk through the working principle step by step. First, there's connection and parameter setting. The user initiates the process via the MATLAB guy on a computer or tablet, wirelessly connecting to the BLE module in the stimulator. The GI provides a search function to find nearby devices and establish a stable connection.
Okay, so they connect wirelessly. Simple enough. Then what?
Precisely. Next is data transfer. Once connected, the GUI sends the desired stimulation parameters, things like the specific current amplitude, the frequency of the pulses, and which electrode pair should be active to the BLE module. The BLE module then efficiently transfers this data serially to the microcontroller using a universal asynchronous receiver transmitter, or UART communication protocol.
Art.
Think of UART as a standardized, reliable way for different parts of the device to reliably send digital information to each other, bit by bit. It's a common method in electronics. The microcontroller then performs decoding and IDAC configuration. It decodes the received information and configures its internal IDAC block. For stimulating the brain, it's typically set to current sync mode to generate the specified current amplitude. The values for things like the anodic and cathodic pulse on times, the interphase delay, and the number of pulses per train are often predetermined and hard-coded into the microcontroller based on established experimental protocols, like the 200 microseconds for pulse on times and a 100 microsecond interphase delay mentioned in the source.
So some settings are fixed in the code, others are adjusted live.
Exactly, based on what needs flexibility and what can be standardized for a given protocol.
Okay. And then it knows where to send that current.
Right. Following this, there's electrode pair activation. Based on the electrode pair chosen by the user via the GUI, for example, pair 1, connecting electrodes E1 and E2, specific general-purpose input-output, or GPIO, pins on the microcontroller control the select lines of the SPDT switches.
GPIO pins, like little switches controlled by the microcontroller.
Exactly. These GPIO pins are versatile digital switches or sensors, allowing the device's brain to control other components, like those analog switches, thereby configuring the exact current path. The system is designed such that typically one pair can be active at a single instance, but its architecture actually supports up to 6 electrodes across 4 pairs, with the potential to stimulate 2 pairs simultaneously if needed, though the validation focused on 1 pair.
Okay, path selected. Now the actual zap.
Hey, well, the controlled pulse. The first phase is cathodic pulse generation. Initially, the SPDT switches are configured to create a closed path for current flow from the supply voltage, VDD, through the brain tissue and then to ground. Once the IDS block starts generating its constant current, this current is mirrored and flows from one electrode, say E2, to the other, E1, for that specific cathodic pulse on time. Again, often hard-coded, perhaps 200 microseconds.
Okay, so current flows one way for a tiny fraction of a second, then it reverses.
Precisely, but not immediately. After this, there's a very brief but crucial interphase delay. Following the cathodic pulse, the IDD block is temporarily disabled for a short interphase delay, typically 100 microseconds. During this brief pause, the SPDT switches rapidly reconfigure their positions to reverse the direction of current flow. A specific switch, S5, is even employed to bypass any transient electrical spikes or noise that might arise during this rapid switching process, ensuring a clean and precise signal delivery.
Little pause and clean up before flipping direction. Smart.
Very. This leads directly to anodic pulse generation. Once the switches have reconfigured, the ID8 block is re-enabled. The mirrored current now flows in the opposite direction, for example, from E1 back to E2 for the anodic pulse on time, which would be another 200 microseconds, perfectly mirroring the cathodic phase in duration and amplitude.
Equal and opposite. That's the key for balance. Right.
Exactly. That equal and opposite current flow is fundamental to charge neutrality. And crucially, after this comes charge neutrality maintenance. Following the anodic pulse, the IDACI block is disabled again for the interpulse interval. This interval is calculated from the total pulse period. For example, if the frequency is 500 Hz, the period is 2,000 microseconds. You subtract the 200 microsecond on times and the 5 microsecond delay, leaving 1,500 microseconds. During this vital interval, the electrodes E1 and E2 in our example are actively pulled to ground. This mechanism physically discharges any residual or accumulated charge, guaranteeing charge neutrality for each and every cycle, critically preventing any net charge accumulation that could lead to tissue damage. It's like hitting a reset button after every single pulse pair to ensure safety.
Okay, that grounding step is super important then.
Absolutely essential. Finally, the stimulation protocol continues. The entire biphasic pulse generation sequence repeats according to the programmed frequency, like 500 times a second if it's 500 Hz. The system is typically coded to stop after a set number of continuous pulse trains, say 10, following established experimental protocols, before maybe pausing and starting again or stopping completely based on the experimental design. This complete cycle ensures precise, safe, and controlled stimulation.
That's incredibly intricate. All that precise timing and switching happening in milliseconds to keep the brain safe while stimulating it. The foresight to build an active charge balancing is a core design principle rather than just hoping it balances out. That's truly remarkable. It's not just about stimulating, it's about stimulating responsibly.
Precisely. It reflects a deep understanding of the electrophysiological requirements for safe and effective neural stimulation.
So the design is brilliant on paper, a real marvel of miniaturized electronics and clever programming. But how do they prove it actually works, in the real world? Let's talk about the fabrication, the rigorous testing they put it through, and what they found when they ultimately deployed this system.
Right, the validation phase. This is where the theoretical design meets real-world application, and it's absolutely essential to demonstrate both the system's electrical integrity, meaning it performs as expected electronically, and its biological efficacy. Does it actually do something meaningful in a living system? This is where innovation truly proves its worth, moving from concept to concrete, reproducible results.
First, let's talk about the physical build, the fabricating details. What were the specifics of bringing this tiny marvel to life, making sure it was robust enough for experimental use in, well, a rat backpack?
Hey, right. The PCD, the printed circuit board, was meticulously designed using Altium Designer, specifically version 23. It was fabricated as a two-layer board using an FR4 substrate, which is a common, robust fiberglass material with copper layers on top and bottom for the circuits. The base copper thickness was specified at 0.5 ounces per square foot, translating to 18 micrometers. This allows for efficient current conduction in a small area. To achieve that compact footprint of 4.3 centimeters by 3.8 centimeters, while still adhering to standard manufacturing capabilities, they maintained a minimum track width of 0.254 millimeters, or 10 mils.
Tiny wires on the board.
Very tiny, and a minimum clearance between tracks of 0.18 millimeters, or 7 mils. These fine tolerances are crucial for packing complex circuitry into such a small space without electrical interference or short circuits. Crucially, the PCB assembly placing all the tiny components onto the board was carried out in-house. This gave them direct control over quality and allowed for rapid iteration if needed. And importantly, it was followed by rigorous benchtop testing before any deployment on an animal model.
Okay, so they built it carefully. What about those initial benchtop tests? How do you simulate something as complex and delicate as brain tissue to make sure your signals are clean and accurate without actually touching a living brain? That sounds like a challenge in itself.
Yeah, you can't just hook it up to anything. They employed a well-established and clever method to mimic the Brown's electrical properties. They used an electrode interface board, an EIB, just to easily access the 6 potential electrode terminals, E1 through E6. To simulate brain impedance, they connected a parallel combination of a resistor, specifically 1.95 kiloohms, and a capacitor, 0.01 microfarads, across electrode terminals E1 and E2.
A resistor and capacitor together. Why that combo?
Because brain tissue isn't just resistive, it also has capacitive properties. This specific combination is based on established protocols found in scientific literature for accurately mimicking the electrical load that brain tissue presents. It gives them a realistic but controlled electrical environment to test the device.
Okay, so they built a fake brain load. Then what did they test?
They systematically tested various parameters. The supply voltage, VDD, was set to 5V. Both cathodic and anodic pulse-on times were 200 microseconds. The interphase delay was 100 microseconds. The pulse frequency was 500 hertz. They used 12 pulses per train with a pulse train period of 1 second and a total stimulation time of 10 seconds. Pretty specific protocol. They then varied the current systematically from 100 microamperes up to 1200 microamperes, or 1.2 mNa, in 100 microampere steps, meticulously observing the output waveforms on a digital oscilloscope.
And what did they find? Did it work as expected?
Yes. Key finding one from these tests was that the generated biphasic waveforms were successfully observed across both a purely resistive load and the more realistic resistive capacitive load. And this was true for various current amplitudes. The paper shows an example for 1 mA current amplitude, and it clearly showed the expected balanced square waveform, confirming the device's fundamental electrical functionality. It produced the signal it was designed to produce.
Okay, good start. Any other key findings from the bench?
Yes. Key finding 2 related to accuracy versus functionality. They noted a percentage change, or deviation, between the expected current, what the IDAC was programmed to deliver, and the actual mirrored current that came out. This deviation was within plus or minus 8.38%.
8% difference. Is that okay?
Well, it might sound like a bit, but it's inherent to the type of basic current mirror configuration they used. What's important here is their reasoning. This minor deviation was deemed acceptable for this specific application because the primary outcome of the experiment, the elicited forelimb movement in a live animal, was a main concern, not absolute current precision down to the last microampere. For many biological experiments, a precise and consistent relative change in current, rather than absolute microampere level exactness, is often sufficient to observe effects. It represents a pragmatic and intelligent trade-off. It was good enough for the intended biological purpose.
Okay, that makes sense. It worked reliably, even if not perfectly precisely, on the bench. Now, for the truly critical part, putting it into a live animal model. How do they validate its functionality in actual rats? This is where the rubber truly meets the road, showing it can actually interact with a living brain.
Indeed, this is where the biological relevance comes into play, demonstrating the device's ability to actually do what it's designed for in a living system. This step is indispensable for proving its utility for neuroscience research and showing its potential for future clinical applications.
So how did they approach the animal experiments responsibly, I hope?
Absolutely. Their experimental methodology in vivo began with a crucial step: ethical clearance. They obtained ethical clearance from the Institutional Animal Ethics Committee, reference CAF Ethics 849-2021, prior to conducting the study. This is a standard and vital requirement in any animal research, ensuring that all procedures are conducted humanely and follow strict guidelines for animal welfare. It underscores their commitment to responsible research practices.
Good. So what was the specific goal of the rat experiment?
The study goal was quite specific and well-defined: to deploy a stimulator in a rat model to identify and stimulate the forelimb region in the rat's brain. The hypothesis was straightforward. If the correct brain region was electrically excited, a corresponding forelimb response, like a twitch or movement of the rat's paw, would be observed, even while the rat was under anesthesia. This provides a direct, observable, and quantifiable validation of the stimulator's function in a live biological system.
Okay, a clear test. How did they perform the surgical procedure?
Neurosurgery was performed on 3 anesthetized Wistar rats. Wistar rats are a common laboratory strain known for being docile and consistent, making them ideal for such studies. A small burr hole, essentially a tiny drill hole, was created in the skull to precisely insert a PFA-coated tungsten wire. This wire acts as the stimulating electrode. It had a very small exposed tip, just 127 micrometers, and an overall diameter of 202 micrometers.
As you wire, where did they put it?
Into the motor cortex region. The insertion site was meticulously located using a standard rat brain atlas, like a map of the rat brain. They aimed for 2 millimeters anterior and 3 millimeters lateral from bregma, which is a specific anatomical landmark on the skull. And the wire was lowered carefully to a depth of 1.8 millimeters from the cortical surface. This precision ensures they are targeting the specific brain region known to be responsible for forelimb movement.
Okay, precise targeting. And how was the electrode setup configured with the device?
The tungsten wire, the stimulating electrode, was connected to the E1 node on the device via an electrode interface board, the EIB. A small 1 mm diameter stainless steel screw served as a ground or reference electrode. This was placed in the cerebellum area and connected to the E2 node via the EIB. So the brain effectively became the electrical load connected across pair 1 of the stimulator.
Clever. And they monitored the current, too.
Yes, importantly, a 1 kilo ohm resistor was connected in series with the brain circuit. This clever addition allowed for real-time monitoring of the actual current pulses going through the brain tissue on an oscilloscope during stimulation. It provides immediate feedback on the current delivery.
Nice check. Any visual cues on the device itself?
Yes. The system also included simple visual indicators, a green LED to confirm the PCB was powered on, and a blue LED to clearly indicate when the stimulation phase was actively ongoing. Just helpful visual feedback for the researchers during experiments.
Okay, so they were all set up. What was the stimulation protocol they used in the rats?
They selected pair 1 via the GOI, connecting the motor cortex electrode and the reference screw. They set the frequency to 300 Hz.
300 hertz. Why that frequency?
This specific frequency was chosen because it's a known threshold, based on previous research they cited, to consistently elicit forelimb movement in rats. They weren't guessing, they were using established knowledge. Then the current amplitude was systematically varied, starting low at 30 microamperes and increasing up to 120 microamperes. This allowed them to observe a dose-dependent response, seeing how the strength of the stimulation correlated with the strength of the movement.
And did it work? What were the key findings for biological validation? Did the rats' paws move?
They did. The findings were clear and consistent across the animals tested. Researchers observed a mild forelimb response even at the low current amplitude of 30 microamperes, indicating successful stimulation of the target region.
Okay, so it worked even at low current.
Yes. And as the current increased, the movement swing also visibly increased. This demonstrated a clear dose-dependent effect. Just as predicted, stronger current resulted in stronger movement up to a certain point. The movement then appeared to saturate after 80 microamperes, meaning further increases in current didn't produce a significantly stronger response, suggesting they had reached the maximum physiological activation level for that pathway with this stimulation.
And this matched previous studies.
Exactly. These findings were highly consistent with previously reported work using other stimulation methods, which unequivocally validated the proper functioning of this novel wireless biphasic pulse generator in a living biological system. It showed not just that it works electronically, but that it works as expected biologically, interacting with the brain to produce a predictable outcome.
That's powerful stuff. Not just showing it works electronically on a bench, but that it elicits the expected biological response in a living animal. And the imagery of a rat with this tiny custom backpack stimulator enabling behavioral studies. Even if those detailed behavioral studies are beyond the scope of this specific paper, it really paints a vivid picture of future possibilities for neuroscience. This is truly where the science gets exciting.
Indeed. The system really opens the door for a new generation of behavioral studies. It allows us to correlate neural activity with free, uninhibited movement in a way that tethered systems simply cannot. It removes a significant experimental constraint, which ultimately leads to more ecologically valid, more natural, and more insightful behavioral data. It brings us closer to understanding complex human conditions like Parkinson's.
So with all of this incredible innovation and validation, what does this all mean for the future of brain research and even potential treatments for debilitating conditions? Where do the researchers see this technology going?
The conclusion drawn by the researchers points to several incredibly exciting avenues for the future. The device is validated for its core function: wireless, programmable, charge-neutral brain stimulation in a small animal. That's established. But they are already thinking well beyond this initial deployment, envisioning how to push its capabilities even further.
Like what? What upgrades are they thinking about?
Well, firstly, regarding GUI upgrades, the graphical user interface, which is already quite robust, can be significantly upgraded. They envision allowing real-time programming of other stimulation parameters beyond just current amplitude and frequency.
What other parameters?
Things like the precise duration of the cathodic and anodic pulse-on times, the length of the interphase delay, the specific number of pulses within each train, and the overall pulse train period or timing.
Wow, so you could really fine-tune the exact shape of the electrical signal.
Exactly. Imagine the granularity of control. A researcher could precisely tailor the exact shape and timing of each electrical pulse. This level of control would offer even finer and more nuanced command over the stimulation protocols. It would allow for more complex, adaptive, and tailored experimental designs, which could be critical for, say, mimicking the dynamic patterns of neural activity seen during natural movements or specific cognitive tasks.
That level of fine-tuning sounds revolutionary. And what about stimulating more than just 2 points at a time? You mentioned it had the potential.
Absolutely. Secondly, there's the potential for expanded multi-region stimulation. With the other IDACs that are still available on the microcontroller, the system could be enhanced to generate biphasic current pulses with different magnitudes simultaneously.
Different amounts of current to different places at the same time.
Precisely. This is a huge leap forward. It means researchers could stimulate multiple distinct brain regions at the same time, each with its own precisely tailored stimulation profile. For example, as you suggested, they could stimulate the motor cortex while simultaneously modulating activity in the basal ganglia, two critical areas involved in Parkinson's disease. This capability would allow scientists to explore how different brain regions interact in complex networks during specific behaviors or in disease states, providing an unprecedented view into brain connectivity and function. We could start asking much more sophisticated questions about how neural circuits truly operate.
That's where the real complexity of the brain starts to perhaps unravel a bit more. And finally, and this directly ties back to the initial clinical motivation, they envision broader applications in disease models.
Indeed. The team explicitly envisages upgrading the system to stimulate both surface and deep brain targets with variable current amplitudes, specifically for use in Parkinsonian rat models. This directly addresses the initial clinical need for better deep brain stimulation outcomes for human patients. This tool could be used to precisely map which deep brain structures, or even specific subregions within them, respond best to stimulation for different Parkinsonian symptoms in the animal model.
So it could help figure out exactly where to stimulate in humans.
Potentially, yes. It could help refine the optimal placement of electrodes in future human DBS, leading to more targeted and effective therapies. Furthermore, this device could be instrumental in understanding the progression of neurodegenerative diseases. By applying specific stimulation patterns over time, researchers might be able to see if they can slow down or even reverse certain pathological processes in animal models. Or they could test the efficacy of novel drugs when combined with electrical stimulation. It's not just about improving existing treatments, it's about potentially pioneering entirely new therapeutic strategies.
What a deep dive. Seriously. We've journeyed all the way from the pressing clinical challenges of Parkinson's disease treatment through the intricate electronic components of a tiny wireless brain stimulator, and then all the way to its successful validation in live rat models. This team has truly created a powerful new tool for neuroscientists worldwide. It's really impressive. We've seen how they brilliantly tackled problems like bulky equipment, those restrictive wires, and rigid, inflexible programming. Their solution offers a device that's small, incredibly lightweight, programmable in real time, ensures tissue safety through active charge neutrality, and can even stimulate multiple brain regions simultaneously. It's just such an elegant solution to a host of complex problems, promising to accelerate discovery in an incredibly important field.
Absolutely. And if we connect this back to the bigger picture, this research provides a fantastic example of how engineering innovation directly fuels scientific discovery in highly complex fields like neuroscience. The ability to precisely control stimulation parameters in a freely moving animal, combined with a quick development cycle using off-the-shelf parts. It just accelerates the pace at which we can unravel the mysteries of the brain. Ultimately, it brings us closer to understanding and treating devastating neurological disorders.
So what stands out most to you about this incredible piece of technology, having gone through the details?
For me, it's probably the integration, taking all those different needs, small size, wireless, programmable, safe, multi-output, fast development, and finding a way to address them all using readily available components. It's a testament to really clever practical engineering meeting a clear scientific need. The charge neutrality aspect is also fundamentally important for ethical and valid science.
Yeah, that integration is key. This whole deep dive really makes you think about the future, doesn't it? Beyond just improving existing treatments for Parkinson's, what could real-time, untethered, precise brain stimulation mean? Not just for understanding neurological diseases, but maybe for unlocking other mysteries of the brain we haven't even considered yet. Imagine the new questions we can start asking, the new frontiers we can explore when our tools become this advanced and flexible. What else might we learn about memory or decision-making or consciousness itself when we can interact with the brain in this way?